Hello, I'm Chaitanya Acharya from MMRF.
Hi, I'm George Mulligan with the MMRF as well. We're in the IMS conference, annual conference here in Rio in Brazil. We had a talk this morning that covered some of our immune atlas research. Chuck, maybe you can share what that talk was about?
Sure, yeah. Our immune atlas is just building upon what CoMMpass has given us for so long. And as many know, CoMMpass is a longitudinal observation study. It's a decade-long study. And we've built a large body of work based off of CoMMpass by looking at tumor genomics of newly diagnosed multiple myeloma patients, about 1,142 of them. And our goal of this study is to describe the immune composition of the bone marrow microenvironment of these newly diagnosed patients enrolled in CoMMpass. So to that end, we obtained bone marrow aspirates from these patients, isolated only the tumor microenvironment part, and we assayed them. And we obtained a single cell map of the immune microenvironment of all the newly diagnosed multiple myeloma patients. And we discovered some fascinating things associated with the biology of newly diagnosed patients, and we described them in detail in our talk at IMS.
Great. So, Chuck, maybe you can comment around parts of the outcome of the study with regard to the clinical results from the patients.
Yeah, so we described the biology, single-cell biology, from the point of view of the outcomes on these newly diagnosed patients, and we categorize these patients into progressors and non-progressors. So these rapid progressors are patients that have had a progression event following therapy within the first 18 months, and these non-progressors have not had any progression event for 4 years at least. So when we looked at the immune composition between these two groups of patients or subjects, we've noticed marginal differences in the cell composition, particularly in monocytes and T-cells, that led us to deep dive into individual cell compartments, for example, CD8 T-cell compartment, and we observed that these rapid progressors seem to be having accumulation of highly dysfunctional CD8 effector T-cells, which was a new finding given the scale of the study.
So that's really interesting. I think another part of the study that is fascinating is that patients were followed for their clinical outcome, progression and overall survival, for 8 years or more. And in this setting, they're being treated with common therapies that are used in between 2014 and 2020. And yet still, can you say more about what was observed with regard to clinical outcome and progression?
Yeah, so with respect to, again, going back to rapid progressors, there is an accumulation of these inflammatory phenotype of monocytes, and there is a strong interaction between T cells and monocytes that seem to be progressing rapidly in rapid progressors. And this interaction is happening between very specific compartments of T cells and monocytes, and that's something that was not previously observed. But again, given 1 million cells in this single cell atlas that we have mapped so far, enables us to understand these interactions and gain more insights into why some patients are progressing and some patients are not progressing, at least from the tumor microenvironment point of view.
And maybe another detail around the association with known risk factors like cytogenetics. Is this information redundant with that, or can you say it's complementary?
Yeah, it is actually, it's a good point that we are trying to make, again, in an effort to understand the biology driving the outcomes in myeloma. We looked at known factors that we can make use of to describe high-risk patients and standard risk patients. And when we combined the signatures that we obtained from our immune atlas with the risk stratification, then we are able to better stratify the patients. It seems to me that there's this complementary action happening between tumor microenvironment and this classical cytogenetics that define high-risk myeloma.